Okay, in this topic, we're going to look at genetic technologies and cloning. We'll spend the first hour of this topic covering the basis of genetic technologies and cloning, and then eventually we'll go on to do some exam questions like we normally do. So we're going to look at what cloning is, understand the uses and disadvantages of cloning animals and plants, learn about tissue culture, and learn the uses of therapeutic cloning and its advantages and disadvantages. So first of all, what is cloning? Cloning is basically just the production of identical copies of organisms, cells, or DNA. So it doesn't have to be of a whole organism, it can just be of cells, so we can clone cells, or we can clone DNA. The definition of a clone is it is a genetically identical organism or a genetically identical group of genetically identical cells derived from a single parent. Cloning does occur in nature, so often we assume cloning is this sort of sci-fi thing of cloning of sheep and humans, but it does occur in nature as part of growth and reproduction. And this natural process can be manipulated to produce clones of organisms artificially, but lots of plants do, and other organisms do clone naturally in nature as part of their growth and reproduction cycles. The scientists are able actually to use genetic technologies to clone whole organisms. And when we talk about this, we call this reproductive cloning. Farm animals or crop plants that got really, really good features, so like cows that maybe provide a good amount of beef, or crop plants that are resistant to certain strains of bacteria or viruses or pests. So they're really desirable, could be cloned. And they're really great because they could ensure a constant food supply and really high productivity. So it's thought that reproductive cloning could really help to increase the number of rare or endangered species as well. So animals that could risk going extinct, we could use reproductive cloning for those. So we have actually done some reproductive cloning of animals, and we're going to look at the process of this. So we can produce artificial clones of animals, and artificial cloning can be used to produce animals that have identical DNA. And we do this by somatic cell nuclear transfer. And we're going to look at how this works, and we're going to look at Dolly, the sheep. And this was done at the Roslin Institution. So Dolly, well, there was a thin Dorset donor ewe, and what happened was mammary gland cells were taken from the udder of this thin Dorset sheep. So the scientists came along, took those mammary gland cells from the udder of this thin Dorset sheep. These somatic cells, somatic just means body cells, adult somatic cells, body cells, they are cells that have been gone through specialization, they've gone through, they're specialized cells. They were placed in low nutrient growth medium. And the reason this was done was to prevent them from dividing and differentiating, but they were still living. So it didn't kill them, it just meant they were not dividing and they weren't differentiating. So these mammary glands were taken and put into that low nutrient growth medium. Then egg cells were then also taken from a Scottish black-face ewe, and the nucleus was removed from these cells. When we remove a nucleus from a cell, we call them enucleated cells. So the egg cells were taken and we removed the nucleus from them. What then happened was these cells were subjected to impulses of electrical current. And when this happened, and they were both placed in the same location, this caused the two cell types to fuse. And what this did is it produced a cell containing the cytoplasm from one sheep and the nucleus from the other. So basically we had the egg cell that had come from black-faced ewe, the cytoplasm from that ewe, but it's actually got the nucleus that had come from the sheep. They produced 277 fused cells, and of these fused cells, 29 cell fusions actually developed into early embryos. So then they took these early embryos and they implanted them into 13 Scottish black-faced sheep, and they acted as surrogates in this situation. Only one pregnancy went to full term, and this was Dolly the sheep. Now, you might wonder why they were using all these different coloured sheep. The reason for this was to prove quite easily, even without doing the genetics, which they would do, that this sheep was a clone of this sheep and not the other two. It looked nothing like the others. It wasn't a natural pregnancy, it wasn't natural fertilization. It was actually cloning using a surrogate sheep. So, somatic cell nuclear transfer, true or false? An egg cell is taken from the organism that is to be cloned. That is false. We don't take the egg cell from the organism that is to be cloned. The two cells are fused using pulse of electric current. That is true. The early sheep embryos were implanted into their biological parent. That is false. Nope, that is not true. That is false. Jolly's genetic material was obtained from an udder cell. That is true. Somatic cells are placed on a low nutrient growth medium to prevent them from differentiating. That is true. Why is it saying that's false? It's not, it's true. Right, so some plants are able to naturally produce genetically identical offspring. We call them, when we've talked about stem cells before, totipotent. And that means each cell has the capacity to regenerate the entire plant. So adult plant cells are totipotent, and from one of those cells, they can regenerate the entire plant. So for example, the English elm tree, and it can be reproduced by something called vegetative propagation. And this is asexual reproduction, and that involves producing new plants from existing vegetative structures. So root suckers or basal sprouts can form from the roots of elm trees when they're under stress. So asexual reproduction usually only occurs so some plants can go through sexual or asexual reproduction. Sexual reproduction is favourable because it increases genetic diversity and a larger gene pool. But in certain conditions where sexual reproduction isn't able to go ahead, and the plant's under stress and it could die, then asexual reproduction occurs, and that's when things like root suckers or basal sprouts can form to allow this asexual reproduction to occur. So this happens, for example, like this. So where the tree can't withstand its current environmental conditions, it still has a chance of survival because these basal sprouts can allow the organism to regrow meters from the original tree, so they can sprout out and it can regrow somewhere else. Large-scale cloning of plants, so more human cloning of plants. A plant that's got highly desirable characteristics, like I was saying, high yield crop plant, or might be resistant to certain pests, can be used to produce more plants with exactly the same genetic composition. A large number of cloned plants can be propagated really easily because all those clones require exactly the same conditions. They will grow at exactly the same rate and will therefore be ready for harvest at the same time. So it reduces costs greatly. It's not like this plant's ready and then a couple of weeks later this one will be ready. Everything will be ready at the same time. So the production of plant clones can occur at any time of year. So how do we do this? Well, the production of artificial clones from tissue culture we call micropropagation, and it's a commonly used method of tissue culture. And it can produce us a large number of plants from a really, really small amount of plant tissue. So what we do straight away is we get a small part of the plant tissue and we remove it from a shoot tip of the plant to be cloned. And we call this tissue an explant. So we remove a tiny, tiny bit of tissue from the shoot tip, and that is an explant. And this explant is then grown on a nutrient growth medium. And each plant cell divides, but the cells do not differentiate into cell types. Each forms a mass of undifferentiated cells, and we call this a callus. Once the callus reaches a certain size, it is grown on two different growth mediums. The first one will contain a hormone that encourages shoot growth, and the second contains hormones that encourage root growth. Once these plants have reached a suitable size, they are then transferred to soil where they can continue to grow. So the sequence of events, an explant is obtained. The tissue grows into an undifferentiated mass of cells, which is called a callus. And the callus is grown with a hormone promoting shoot growth. The callus is also grown with a hormone promoting root growth. And then the plant is transferred to soil. So this sounds really great. We can make loads of money from this. It really helps us with crop growth, with animals, but there's actually disadvantages of reproductive cloning. A population of cloned individuals will actually have a really, really low genetic diversity. This reduces the ability of that population to adapt to any changes in the environment. So clones will equally be vulnerable to a disease or pests. So those diseases spread a lot faster and they will affect the whole population. So a great example of this is Dutch elm disease, and this was accidentally spread through Europe, and it was a disease that devastated native elm populations. And it is thought that these were natural clones from only a few individuals. So it wasn't ones that we'd gone and cloned ourselves, but they'd naturally cloned themselves. The first cloned mammal, Dolly the sheep, actually died really early due to lung disease. And this actually raised concerns about the health and life expectancy of cloned animals because the original version of Dolly didn't die prematurely due to lung disease. So it wasn't like a genetic thing that had been passed on. Some cloned mammals have telomeres of different lengths compared to normal animals of the same species. And Dolly had shorter telomeres. And what telomeres are, they're pieces of non-coding DNA. And when we've talked about that before, they're on the ends of our chromosomes, and they prevent our chromosomes from degrading. And every time our cells divide, the telomere shortens, so we lose some of the telomere. And the more our DNA replicates, the more those telomeres get broken down. And then eventually you affect the DNA that's there. So we can measure the aging of cells by the length of telomeres. So someone who is older would have shorter telomeres in their cells than somebody who's younger. If you automatically have shorter telomeres, your chromosomes are going to degrade quicker. It could be that obviously the original sheep had shorter telomeres because it was an adult, and then we've got a baby being grown with adult length telomeres. So obviously as eggs are going and then embryos are going through cell division constantly during reproduction and then growth and cell differentiation in gestation, those telomeres are going to be shortening because we have so much cell division during that period of time. So by the time that we get a full sheep, and those telomeres could be really super short, which then could lead to these issues such as lung disease. We can have something called therapeutic cloning. So organs, tissues, and cells can also be produced using cloning techniques. So this is known as therapeutic or non-reproductive cloning. So we're not making a whole new organism, we're just making new organs, tissues, and cells. So we're going to look at what it is, some good things about it, and then some bad things about it. So the technique is still somatic cell nuclear transfer, and is used to produce an embryo containing genetic material from a somatic cell. So the process at the beginning is very, very similar to the sheep. But what we do is we get pluripotent embryonic stem cells are also extracted from the embryo. And these cells are capable of differentiating into all types of cell in a developed organism. And we can use these to produce a desired cell, tissue, or organ. So we get that embryo, we get it growing, take those stem cells out, and then we grow the desired tissues, cells, or organs that we want rather than a whole organism. So some advantages of this is that therapeutic cloning has the potential to produce cells, tissues, and organs to replace those that have been damaged either by disease or accidents. Using these cloned cells have some great advantages because we could actually take cells from the patient and create them from them. Because they're identical to the patient, it prevents the risk of rejection, so somebody wouldn't have to take drugs for the rest of life like they do when they have donor organ. They have to take anti-rejection drugs. You wouldn't have to. It also alleviates the current problem of donor transplant waiting lists. These are very, very long. They can be years long. And sometimes some people just never get the organs that they're waiting for. And it could also be possible to treat some diseases or damage that we can't currently be repaired by transplantation. And that's due to the totipotency of embryonic stem cells. So we can actually replace some things that maybe, because if we could put the stem cells in where that damage is, we could then maybe hope that those stem cells would differentiate into the correct type of cells that would be required in that area in places that you couldn't do a traditional transplant. However, there is disadvantages. Once we've extracted those stem cells from the embryo is then destroyed. And some people believe that that's morally wrong to destroy these early embryos as they are a potential human life. Another problem with therapeutic cloning techniques includes the difficulty to produce actually a viable egg, and it can take hundreds of attempts to fuse cells before a stable egg is actually formed. In mature adults, we only have a few stem cells remain, and these aren't totipotent or pluripotent, they're only multipotent. And they have the capacity to only differentiate into only a few specific cell types. So they maintain and repair specific tissues in the body. So we have some in our bodies, in blood cells, for example, in bone marrow. So adult stem cells do not provide the same flexibility as embryonic stem cells. However, we can use them to produce a limited range of tissues for transplantation. So adult hematopoietic stem cells, we find them in bone marrow, and we've been using them in transplants for 40 years, and these stem cells form all the blood cell types in the body. So if we've got any issues with bloods, we can do them stem cell transplants, which is a blood stem cell transplant, so bone marrow transplant basically. So patients with something like leukemia or other blood cancers can be treated using that.